Hello, dear all. Welcome back to the channel of Curious Corners in Biology. After a brief pause, because I had some commitments. So, as usual, this is a very interesting channel to discuss anything and everything about biology, anything and everything which can kindle interest in the minds of learners of any age or any background. So, today, I have come up with more questions from respiratory physiology, and we will also be looking into some interesting questions from hematology. And please subscribe to this channel if you like such interesting questions, inquisitive questions. Please hit the bell icon. And I have written a book, Curious Corners in Biology, the link of which is given in the description. So please check that out. That's available on Amazon Kindle. So let us start with respiratory physiology. So you might be able to see my screen. Just a minute. Yeah. So I hope the screen is pretty much visible. So the first question of respiratory physiology is about oxygen dissociation curve. So first of all, let us understand in brief what is oxygen dissociation curve. This curve is simply a graphical representation about how much oxygen is bound to hemoglobin, because the function of hemoglobin is to transport oxygen and carbon dioxide. What hemoglobin does is it delivers oxygen to the tissues. Oxygen is used up by the cells of the tissues to produce energy and release carbon dioxide. So here we understand one thing, that when hemoglobin binds with oxygen, it transports oxygen and then it also has to release oxygen to the tissues. Okay. So how does this take place? So in here, there is a chemical intermediate which is produced by RBCs in our body, which is called as 2,3-biphosphoglycerate. This 2,3-biphosphoglycerate has a role to stabilize the structure of hemoglobin without oxygen. Hemoglobin has natural affinity to bind with oxygen, and it forms oxyhemoglobin after binding with oxygen. But then hemoglobin also has to release oxygen to the tissues. That means there should be some trigger for hemoglobin to dissociate with oxygen, to separate from oxygen, so that oxygen can reach to tissues. And this trigger is 2,3-biphosphoglycerate. So, by this graph, we can understand that in presence of 2,3-biphosphoglycerate in RBCs, oxygen simply separates from oxyhemoglobin. That means hemoglobin will unload oxygen to the tissues. And when oxygen binds to hemoglobin, then oxyhemoglobin is formed. Then this curve is shifting towards left. That means oxygen and hemoglobin bind tightly. Hemoglobin is having more affinity to oxygen when this curve is shifting towards left. And it shifts towards right when 2,3-biphosphoglycerate binds with hemoglobin and dissociates oxygen from hemoglobin. So it reduces the affinity of hemoglobin to oxygen, thereby shifting the curve towards right. So this is left shift and right shift of oxygen dissociation curve. So hope you get this concept. Okay. Along the same lines, there is one more interesting question. Actually, there are two types of hemoglobin: adult hemoglobin and fetal hemoglobin. By adult hemoglobin, we mean maternal hemoglobin, and fetal hemoglobin is that which is present in the blood of fetus. So this question says that how does adult hemoglobin differ from fetal hemoglobin? Okay. So first of all, the structure is slightly different of adult hemoglobin from fetal hemoglobin, but mainly fetal hemoglobin has more affinity for oxygen because it binds with 2,3-biphosphoglykoside with less affinity. That means it will not bind 2,3-biphosphoglykoside as good as adult hemoglobin binds. So that is why in normal conditions, fetal hemoglobin has 30% more affinity to oxygen as compared to adult hemoglobin, that is maternal hemoglobin. Now, why this is a requirement? Because of course, fetus grows and develops vigorously. So the demand of oxygen by the fetus is very much high. So, of course, fetal hemoglobin should capture more oxygen in less time and with more affinity as compared to maternal hemoglobin. And this is clearly shown by this graph. This is oxygen hemoglobin dissociation curve of fetal and maternal hemoglobin. So, on the right is maternal hemoglobin curve, and on the left to the maternal hemoglobin is fetal hemoglobin curve. Of course, this curve is going to level off because hemoglobin can be saturated with oxygen. Okay. So here we can see that fetal hemoglobin naturally has more affinity to oxygen. Okay. And it has less affinity to bind with 2,3-biphosphoglykoside, which is reducing the affinity of hemoglobin to bind with oxygen. That is why this is having more affinity to oxygen. So hope this answers the question. Okay. So here we complete questions of respiratory physiology from my side. If you do have questions, any additional thoughts or inputs, any curious questions which you are finding mysterious or unsolvable, please let me know in the comments. We can discuss those questions. And now we are moving to hematology. Hematology is simply study of blood. Okay. So the first question is very simple. This has aroused much curiosity from our childhood. Okay. That how does oxygen affect color of blood? See, we know that blood in arteries is bright red and blood in veins is deep red purple. Okay. Veins, if you see, they are superficial and they are light blue in color if they appear on our hands. Okay. Why so? Why such difference in color? First of all, when blood contains oxygen, then oxygen binds with the iron of the hemoglobin, and iron of the hemoglobin makes the blood in presence of oxygen look bright red in color. While without oxygen, the hemoglobin, that is the iron component of the hemoglobin of the blood, will make it look deep red purple. So, of course, blood which is withdrawn from veins will be deep red purple, and blood which is withdrawn from arteries being oxygenated will be bright red in color. Of course, veins will appear light blue in color not because they are light blue. They are not light blue in color, but simply light is scattered in a way and blood is also flowing through the veins. This phenomenon of scattering of light, especially, and sort of refraction phenomenon makes the veins, which are superficially located, appear light bluish in color. Okay. So this is a very simple question. Now, this is the heart of today's discussion. This is a very good question actually, but it's very simple to answer. We all have heard about blood transfusion. Okay. Blood transfusion is simply transfusing blood, that is transferring blood from donor to recipient. Recipient is the patient who needs the blood for any reason. Okay. But then blood has blood cells, RBCs, WBCs, and platelets. And don't you think cells would have their own DNA? Yes, of course. Then blood transfusion, shouldn't it alter the genetic makeup of the recipient? Yes or no? Does it have any effect on the genetic makeup of the recipient? What is it? What not? Okay. So this is a very interesting question. And what happens to donor DNA? Because when blood is transfused, even after blood matching, what happens to donor DNA? Does it integrate with the recipient's DNA? Does it not integrate? Does it stay there? What happens? So there is a very interesting answer to this. First of all, let us think about the blood cells. We have mainly three kinds of blood cells: RBCs or erythrocytes, WBCs or leukocytes, and platelets or thrombocytes. Which of these cells would have DNA? Only WBCs. Okay, RBCs and platelets don't. So we have to think about what happens to WBCs. Now, when blood transfusion takes place, WBCs from donor's blood also enters the recipient's blood. Okay. So they will remain there. Okay. And this remnant of donor WBCs in normal cases of blood transfusion would be around for few weeks. But if the patient receives large blood transfusion, then the donor WBCs can remain in recipient's blood for as long as 1 or 2 years. But usually transfusions are not that large, so it is only a matter of few weeks that donor cells would remain in recipient's blood. Now, even if we amplify donor DNA using techniques like PCR in recipient's blood, that is polymerase chain reaction, then we also find donor DNA in recipient's blood. This phenomenon is called as microchimerism. Presence of donor DNA after blood transfusion in recipient's blood is called as microchimerism. Now, this is gender independent, whether a male recipient receives from female donor or a female recipient receives from male donor. It doesn't change microchimerism. One of the interesting case is intrauterine blood transfusion. Intrauterine blood transfusion is a necessity in case of hemolytic disease of the newborn. This occurs due to Rh incompatibility when maternal antibodies cross the placental barrier and destroy the RBCs of the newborn, or not newborn, it is fetus only because it occurs at the stage of fetus. So, intrauterine transfusion will cause maternal DNA to stay in the blood of fetus for as good as many years or several years. Why so? Because the immune system of the fetus is not that much developed to recognize self and non-self. So it does not recognize foreign cells. It does not recognize foreign DNA. So in that case, the donor DNA or leukocytes from the mother or from any donor has been seen to remain in fetal blood. Okay. So this answers the question, and of course the genetic makeup does not change because blood transfusion is a somatic phenomenon. Nothing happens at the level of germ cells. Okay. And yes, one more thing, umbilical vein is preferred to transfuse blood. Always remember that veins are always preferred over arteries because veins will have less pressure of the blood flowing. They are having thin walls, they are less elastic. So when blood is injected, it is not going to ooze out and the procedure is less painful. If blood is injected into arteries, arteries will always have more blood pressure than vein. So the pulsatile pressure of artery is also more, and arteries will have more nerves as well. So if arteries will have more nerves, then it can make the procedure more painful as well. Okay. So of course, veins are preferred over arteries. The last question is, can platelet count increase? We have seen that RBC count can increase, that is altitude sickness can decrease in anemia. WBC count can increase or decrease in infections, but what about platelets? Yes, platelet count can also increase, and this is called as reactive thrombocytosis or secondary thrombocytosis. Okay, so what are the conditions of reactive thrombocytosis? Conditions are connective tissue disorders, inflammatory bowel disease, tuberculosis, cancer, and spleen which is absent. Okay, so such conditions can cause the platelet count to increase because whenever a person undergoes surgery or has any infection or inflammation, the platelet is going to increase temporarily. Why temporarily? Because this is not a bone marrow condition. The signal is not from bone marrow. Till the infection or inflammation prevails, the platelet count is increased, and then when the infection clears out or when the wound is healed, then the platelet count will return to normal. Okay. So that much for today. We will see you soon with more questions on hematology. And if you are interested in such curious content, please subscribe to the channel, hit the bell icon, and check out my book Curious Corners in Biology in the link given in the description. Till then, see you. Bye.